Hi everybody, so today we're going to be talking about mitosis and meiosis. So before we actually go into the different cycles themselves, we have to go over some terminology first. So first of all, the definition of homologs is two non-identical copies of a chromosome that come from each parent. So one from dad and one from mom. Sister chromatids are actually two identical copies of a single replicated chromosome that are connected by a centromere. So you'll only see sister chromatids after that chromosome has replicated. Okay, so here we have a pair of homologous chromosomes. The red one would be the one from the mom, so the maternal chromosome, and the blue one would be the one from the dad, so the paternal chromosome. So these chromosomes are going to replicate, and after replication, we will see sister chromatids. Okay, so now this X-like sort of chromosome structure is actually a replicated chromosome. And we can see the sister chromatids are being held together at that centromere, which is that dark circle area. And now the relationship between these two chromosomes we would refer to as a homologous pair of replicated chromosomes. Okay, so let's talk about mitosis in a 2n=4 cell just for the sake of simplicity. So the cell actually spends most of its life just hanging out in interphase. Prior to actually going into mitosis, the cell will replicate its DNA, and note that at this stage, the DNA is still uncondensed, it's quite loose, and we've indicated that with these loose squiggles. So when that DNA is starting to be condensed, that's the start of prophase. And during prophase, essentially, the DNA condenses, and we see these chromosomes starting to form, and also the spindle is starting to form, and you can see that with those tiny little T-like structures at the poles of the cell. And during metaphase, the chromosomes will actually line up at this metaphase plate that's sort of like the equator of a cell. So in addition to the lining up of these chromosomes, the chromosomes will actually start to be attached to the spindle fibers. And these spindle fibers is essentially what is going to pull apart these chromosomes during anaphase. Okay, so in anaphase we actually see the sister chromatids are being separated by these spindle fibers, and they're being pulled towards opposite ends of the poles of the cell. And so finally we have telophase and cytokinesis. So during telophase, the chromosomes begin to unwind and decondense, and the nuclear envelope will start to reform. And during cytokinesis is the actual formation of the two daughter cells, and note that the two daughter cells are both 2n=4. Okay, so now let's talk about meiosis. In meiosis, we're going to start in interphase as well, and note that the cell is also 2n=4. And the cell will actually condense its DNA just like it does in mitosis. And so here we're going to see some condensed chromosomes, and the key in prophase here is the pairing up of homologs, which we don't see in mitosis. So they form these structures called tetrads with the homologs pairing up. And also during prophase, we would see what's called crossing over. And crossing over is the exchange of genetic material between two homologous chromosomes, and this generates genetic diversity. And following prophase, we have metaphase, and that is when the chromosomes line up at that metaphase plate, just like they do in mitosis, with the exception that in meiosis, they line up in pairs. And just note that the pairs can be in any orientation. So the top chromosome here is shown as a blue-red pair, and then the bottom we have a red-blue pair. So following metaphase, we have anaphase, and just like in mitosis, the chromosomes are being separated and pulled towards the opposite ends of the cell by the spindle fibers. And here we're actually pulling the chromosomes to separate ends of the cells, not chromatids. So that would be a difference between meiosis and mitosis. And so by the end of meiosis 1, we have 2 daughter cells, and the key here is that these daughter cells are already haploid. So these cells are both n equals 2 rather than 2n equals 4. So by the end of meiosis 1, reductive division has already been completed, and we've already gone through the process which gives us genetic diversity or recombination. So now let's look at meiosis 2, which would be the second half of meiosis. So the starting cell here that we're seeing in the prophase 2 stage is actually one of the same cells that we saw at the end of meiosis 1. So we're just going to keep following this one cell. And the final product of meiosis 2 is going to generate 4 unique daughter cells in contrast to mitosis, which generates 2 identical daughter cells. So meiosis 2 is actually quite similar to mitosis because of the lack of pairing of chromosomes and just how the chromosomes will line up evenly at that metaphase plate. And here we are separating the chromatids just like we did in mitosis. Furthermore, there is no reductive division and there's no crossing over happening in meiosis 2. So that's why a lot of people say that meiosis 2 is basically just like mitosis. And at the end of meiosis 2, we end up with 4 haploid daughter cells that are unique. So I just want to leave you guys with this question here. What are 2 major differences between mitosis and meiosis? Thank you for watching. If you guys enjoy this sort of content, make sure to hit the like and subscribe buttons and give us a comment and let us know what you want to see in the future. We'll see you in the next video. Thank you.